Thanks very much. It's always a particular pleasure to be at the RI giving a talk. And today I'm not going to talk about robots taking over the world or toxic waste turning us all into monsters that might look like that. This is going to very much be a talk about the natural world. So are there any mutants in the audience is my first question. Yeah. There are. Of course there are, because actually we're all mutants of one kind or another, because we're all very different from one another. And that's what this talk is all about. It's about the diversity in the natural world amongst humans, but also in all of the species that make up the natural world. And where does it come from, and what does it mean, and where's it taking us? So that's what I want to think about today. So greetings, fellow mutants, is my first message. So what about these dogs? I think that's one of the great examples of huge amounts of natural diversity in a species, in size and shape, much more diverse in many ways than we are. So why is that? And that's because for thousands of years, we humans we've been breeding mutant dogs. They've got a characteristic that's favourable. Once upon a time, that was because we use them to work, and now it's mainly because we want to keep various species going because we like the look of them and we like to own these dogs. And they have a characteristic or a trait that's attractive to us as their human masters. But of course, all of these characteristics, these are mutations. So these are mutant dogs. And when we breed a characteristic, it goes down, it's passed down through the generations. So in effect, we have created this extreme diversity in dogs. Not all of these species would have arisen naturally. And that's something that we're going to be coming on to think about a little bit more. But at the end of the day, even though these species of these varieties of dog look so different, they're all dogs, right? They're all the same species. A dog is still a dog, even though they look so different. And that's because it turns out that actually the differences between these dogs, if you just take one characteristic like the length of the fur or the degree of curliness of the fur, you'll find that it's caused by about 3 mutations. That makes the difference in the appearance of those dogs. So what exactly do we mean by mutation? That's the first thing we need to think about. And in order to understand what we mean by a mutation, we need to go back to this molecule, to DNA. We've all heard of DNA, the thread of life. So this is found in every living thing, as well as every human being. Has a certain beauty, I think, in its simplicity. So we've got long chains of letters, what we would call bases, and they're held together by this strong backbone, sugar and phosphate, if you're into the chemistry, backbone if you're not, A, C, G, and T. And the bases always connect with each other in the same way. So A always pairs with T, and C always pairs with G. And we don't need to worry about the chemistry of those sorts of molecules. But the study of DNA is, I would say, one of the greatest triumphs of modern science. So the overall shape is a double helix. It's a long spiral. It's ordered. It's simple. It's regular. And these 4 bases make up this alphabet of 4 letters that makes up the genetic code. And that's the key. It's a code. A code for holding all the information to make all living things, a cat, a bat, a dinosaur, or a human. So I think that the DNA molecule is the most amazing thing. And I think it's even more amazing when you look at where it is in our body. So it's in our cells. We have about 8,000 or so cells can fit on the head of a pin. We contain about 40 trillion of these cells, and in each of our cells is a nucleus, and in the nucleus are the chromosomes, and the chromosomes are made up of DNA. And so long strings of bases make up instructions for a characteristic like the length of fur on a dog. That's a gene, the DNA that makes a particular characteristic. So that's where DNA is in cells. And then the other really important thing about DNA is that when a cell divides into 2, then that DNA is first copied absolutely, completely, everything is copied, and then it's divvied out absolutely evenly amongst those two daughter cells. And this picture shows how this happens in a cell. So actually, in blue is the DNA, and the green are things called microtubules, and they're like the cytoskeleton. They're the structures that give the cell its shape, and they make sure that when the DNA is copied into two, it's absolutely faithfully segregated into two halves, so that when this cell divides, each daughter cell, we call them daughter cells. I don't know why we don't call them son cells, but we don't. We always call them daughter cells. They get exactly the same copy. So you could say actually that biology is the only science when multiplication and division mean the same thing, right? Because when the cell divides, it multiplies itself into two. And this is a remarkable feat. Each one of our cells, and remember we can get 8,000 cells on the head of a pin, they're really, really tiny, and we've got 40 trillion of them. Each one of those cells, even though they're so tiny, contain about 2 meters of DNA, which is wound up and folded up so small that it can fit into this very, very tiny, tiny cell. So it's extraordinary how it's packaged and how it works, but it does. Okay, and it does manage to be completely faithfully copied and completely faithfully segregated. And the structure of the DNA provides a lot of clues as to how this all works. So when Watson and Crick solved the structure of DNA in the 1950s, they said that it has not escaped our notice. This was in the last line of their paper. It's not escaped our notice that the specific pairing we have postulated suggests a possible copying mechanism for the genetic material. And the way it works is that the double helix kind of unwinds like it's being unzipped when a DNA molecule is copied. The bonds break, new bases come in. The bases know how to pair because there's rules that govern that, and so you end up with 2 strands. So DNA is a perfect self-replicating molecule, and the beauty of this, I think, is in its profound simplicity, and the mechanism was really obvious once the structure was determined. So now we've seen how simply and easily this DNA replication can happen. Let's think about when it happens. It happens every single time a cell divides. Every time any of your body cells divide, then you have to replicate that 2 meters of DNA, unpackage it, replicate it, divide it out, package it back together again. So does anyone know how much DNA they actually make in a day on average? Because you need to replace cells that are being worn out. Maybe if you only have to rub your arm and a load of cells fall off, hundreds, thousands of cells fall off, you have to make new ones. There's all sorts of repair going on in your bodies the whole time. You make about 100 billion meters of DNA every day. That's why you might feel a bit tired sometimes. It's a very good excuse for a nap, if you ask me. And I've sort of impressed on you how well this works. But what we really want to think about now is how does a mutation arise? What if the system isn't so perfect after all? What if mistakes creep in? They needn't be big ones. You could maybe just get the wrong base. Base might fall off. You might just add a few more by accident, extra ones. So a mutation is a change in that DNA sequence. That's the instruction manual for making an organism. In the language of the genes, then, a mutation could change the characteristics of the cell, and that could change the characteristics of a whole organism. If you were a dog, maybe it could make your fur longer, for example. And once a mistake occurs in a DNA molecule in a cell, then it's going to be passed down to every other cell, because we've said that DNA is so faithfully replicated, and so on and so on. It'll be passed down. This is a picture of a fruit fly, in case you were wondering. The head of a fruit fly, an organism that geneticists use to try to understand how genetics works. So that's a normal fly, and this is a mutant fly. Now, can anyone spot the difference? This one has antennae, and this one has legs instead of antennae. I don't know about you, but this to me seems like a big thing to get wrong. Putting the right structure in the wrong place. And this is caused by a mutation, a single base change in one particular really important gene that's required to determine the body plan. So this is an organism being completely reprogrammed. So a tiny change can make a huge difference. Mutations are happening in our DNA all of the time. I don't think you're going to wake up tomorrow morning and find legs growing out of your head. But mutations can happen, and they can have huge effects on the morphology and the functioning of an organism. So when does it matter when mutations happen? In what kind of cells does it really count? Does it matter if mutations happen in our skin cells? Or does it matter more if mutations happen in those cells that go on to make the next generation? Because they're going to be the ones that are passed on. And for the purposes of today's talk, we're going to be thinking about mutations that are passed on. So they matter when they happen in cells like this. This is a human egg that's just been fertilized by some sperm. So these are gametes. Gametes are cells that are going to make the next generation. So if a mutation occurs in cells that give rise to gametes, then those mutations are going to be passed on. That fly with the antennae sticking out of its head, maybe all its babies are going to be just like it. That's going to change the course of that whole family, that whole lineage of flies. So I'm going to show you now some gametes from my favorite organism. I don't actually work on flies. I work on worms, on nematode worms. And it's a great organism to look at because it's see-through, basically. And we can see here the start of life. And this, I love looking at this. Every time I look at this, I think, oh, wow, I really am doing the right thing with my life. So I know that might sound odd to you, but to me, it's really important. So this is an embryo that's just divided into two cells. So that's really early on in the life of anything, just having two cells. But this one down here hasn't even divided once at all. And that's because the male nucleus and the female nucleus, so that, in other words, the nucleus of the egg, the DNA of the egg, and the DNA of the sperm haven't yet got together and fused. And this is what we're watching in this video. So what we'll see are two sort of cleared areas. This is the female DNA here, and this is the male DNA here. And they'll become a bit clearer when I play this video. And they'll find each other. It's very romantic. They'll find each other. Look, there we are, and they fuse together, and that's the beginning of life. And then that embryo is going to divide into two cells, and then those two cells are going to divide into 4 cells, and so on and so forth. So then you can really see that if a mutation happens in these very early cells, that's going to be passed on to all of the cells of that organism, and then the gametes and the next generation, and so on and so forth. Mutations aren't necessarily bad. They don't necessarily make you have legs sprouting out of your head, right? They can do anything. They can be good. They can make you better, more adapted to your environment. And that depends on many things. There's basically no pattern to mutation. That is random. Mutations, they just happen. And they happen for a variety of reasons which we needn't go into, but they just happen. But there is a pattern to inheritance. And the rules of inheritance were first worked out by an Austrian monk called Gregor Mendel. This was long before DNA was discovered. And Mendel was doing lots of crosses of pea plants. And in fact, between 1856 and 1863, he cultivated and tested thousands and thousands of pea plants. And he identified the rules of inheritance. So basically what he found was that characteristics don't blend together like you might imagine they do. So if you take a purple leaf plant and a white leaf plant, all the progeny are purple leaf plants. And the early geneticists thought, well, that means that there's more of a tendency towards purpleness. But when he crosses those purple progeny to each other, he gets the white ones back again. That's the crucial result of Mendel, because it says that inheritance isn't blending, it's particulate. It says that characteristics can survive through generations. You might talk about your grandchildren looking like you. It's skipped a generation. You might say, gosh, all my wonderful characteristics seem to have been missed out in my children. But in my grandchildren, aren't they wonderful? So Mendel's was an amazing finding because actually it was 40 years before anyone had even coined the term gene or genetics. And it was another 40 years even before the naming of DNA as the molecule of inheritance. But his findings were incredibly profound because they provided the rules of how inheritance works. But he didn't, nobody really took much notice of his work at the time. He was a monk hidden away, working away in his monastery in Austria. And nobody really took much notice of it until rather later. But one thing that was obvious around everywhere was this natural variation. I've shown you dogs, these are cows, another one of my favourite organisms. Domestic animals, again, something that we've bred in order to get characteristics that we like in farming. And it was this natural variation, although he didn't understand the rules of it, that was the thing that caught the eye of a young naturalist called Charles Darwin. And this is Darwin. And he went on this voyage to the Galapagos Islands. And when he was there, he went thinking that natural variation was really a cool thing. And when he was in the Galapagos, he found many examples of this, particularly in finches living on different islands. And what Darwin noticed that got him really excited was that finches varied, and it seemed to depend on where they were, which island they were in. And he could correlate the characteristics of these finches with the kind of food that they like to eat. So, for example, the warbler finch here has this long probing beak that you might use for eating insects, whereas this vegetarian finch here has an adapted beak for eating seeds. Looks more like a parrot's beak. And these are the sorts of evidence that Darwin drew on to identify his theory of natural selection. So you could say then that the voyage that Darwin went on was probably the most important voyage ever in the whole history of life on Earth, because it led to this completely revolutionary idea of natural selection. So Darwin said that the variation amongst creatures in their natural environment results in the struggle for life in which only the most suitably adapted are preserved. Now that's one of the great ideas of science. And the idea is modification by descent. So modification is mutation. Now that happens randomly, and it's copied and it goes through generations. Descent is about the rules of inheritance through generations that Mendel discovered. And natural selection then refers to the survival of those variants then that are most suited to their environment. So evolution, it basically means change over time through natural selection. The idea was that organisms change very gradually. Eventually they might gain or lose a limb or a wing or an eye. We've seen Drosophilas that have legs hanging out of their head. I don't think that's an adaptive mutation. I think that could actually be quite bad. But lots of mutations are good and allow organisms to survive in a changing environment. So change is caused very, very gradually by mutations in DNA molecules that are inherited. So change, therefore, is selected for. Some is favoured, some is selected against. And eventually, a new species can evolve. So we can see then where we came from. That's natural selection. Ultimately, one type of creature can be transformed into something completely different. And Darwin's idea of evolution by natural selection was so important that it really provides a unifying theory for biology. But that unifying theory only really works well when you also have a theory of heredity. And Darwin didn't have that. Darwin was very much of the blending inheritance school. And it was Mendel that said, no, it's particulate. But that also works in this theory of evolution. And Theodosius Dobzhansky, when he was writing about Darwin's idea, he said, nothing in biology makes sense except in the light of evolution. And I think that's a really profound statement that's still a very popular idea today. So Darwin's idea then was very bold, it was very brave, it caused a storm in Victorian society, but it's still impossible to overstate the importance of these ideas today. So Darwin thought of this idea of the tree of life, where all species are connected, and this is actually his drawing of the tree of life in his notebook. His tree was a simple one. We now know that it's a lot more complex. We now know that it's a lot more complex than that. And it started off with simple life about 4 billion years ago. And natural selection then has sent organisms off in a myriad of different directions. Mutation fuels that diversity that natural selection can act on. And that gives us the diversity that we have around us today. Animal species on Earth today actually only represent less than 1% of species that have ever existed. That's amazing, isn't it? 99% of species have actually already gone extinct. But now we have 9 million species around us. We have 28 different species of fish. We have 15,000 different species of moss. Who would have thought that? And there are 350,000 different species of beetle. So all of this diversity has descended from a common ancestor some 4 billion years ago. So then it all makes sense then that we are all related to one another. We are about 50% Mexican cave fish. Love these little fish. We're about 70% rat. We're about 97% chimp. So these changes in DNA sequence then have sculpted different organisms, and that gives rise to Darwin's idea of the endless forms, most beautiful and most wonderful, that he talked about. So genes are at the very heart of evolution. So DNA then, it's not just an instruction manual, is it? It's also a history book. It's a historical record of all the genetic mutations that have occurred by chance to make us what we are and to make all living things what they are. So it shows how it's all happened, and it shows how all organisms are related through an unbroken chain of copying of DNA through 4 billion years. So DNA sequences are an incredible historical archive as well as an instruction manual. Imagine how excited Darwin and Mendel would have been if they had known all this when they were proposing their ideas. Now, I'm going to fast forward to the present day and think about the future. We know so much about DNA and genes that we have the technology to actually begin to think about changing our genetic makeup, perhaps even our destiny. And that's what we're going to be thinking about for the last few minutes. So work on DNA sequencing then was started by Fred Sanger in 1977. He'd already developed a method for determining the amino acid sequence of proteins. So this was his second Nobel Prize. He was definitely an overachiever on that front. So he worked out how to work out the base sequence of DNA, and then he scaled up and was able to work out the sequence of quite long stretches of DNA. And then people worked out how to scale up even more and to sequence a whole genome. So that ushered in the field of genomics, the study of whole genomes, all of the DNA that makes up an organism. So the first organism to be sequenced was a bacteria, Haemophilus influenza. That was followed by yeast, so now we're in about 1996. The first multicellular animal to be sequenced was my worm, C. elegans. That was in 1998. And then the fruit fly, a bit behind in 2000. And then the human genome was sorted out in 2003. So this was an extraordinary achievement. It involved hundreds of different workers from around the world. It took about 13 years, cost about $2.7 billion. If you typed out the whole of the genetic code, typing at 200 letters a minute, which I think I could just about do with my 2 fingers that I use for typing, it would take you 29 years, even if you didn't have any lunch breaks. And yet we've got that inside every single one of our tiny 40 trillion cells, and our enzymatic machines can copy it in a matter of just minutes. So genome sequence, then, information brings all of life closer together than was ever thought possible. Thousands of genome sequences are now available, and it means we can interrogate the evolutionary relationships between species in a way with more resolution than we could ever do before. So the fields of biology that gain from DNA sequence, genome sequence information are as diverse as conservation biology, the spread of infectious disease, crop science, and many other areas. What about the medical benefits of genome sequence information? It's easy to be dazzled, I think, by the possibility of using genome information in medicine. Sequencing the genome of your cancer cells may allow oncologists to tailor-make your treatment to increase the chances of success. That's tremendously exciting isn't it? And it's already starting. The speculation then is that healthcare professionals will eventually be able to use genomic information to predict what diseases we might get, or even minimize the risk, maybe even eliminate those diseases altogether through personalized medicine. It's not straightforward, though, this, because human disease is rarely caused by mutations in a single gene, like cystic fibrosis, for example. Most diseases, like heart disease or cancer or mental illness, result from extremely complex patterns of interaction between many, many different types of genes, many different mutations, working together and working with the environment as well. I think there's a public misconception that scientists are discovering the genes for this and that characteristics on a daily basis, that the idea that there's a gene for forgetfulness or binge drinking or intelligence or beauty. Very little in human biology is straightforward at the genetic level. This is just a diagram of all the different pathways that can contribute to one cell becoming cancerous. I don't think genetics is deterministic at all. I think it's better understood as probabilistic. A whole host of genetic factors contribute to the odds, but the outcome is decided by an even greater range of genetic, biological, and environmental influences. So I don't think we need to take a dystopian view of medical genomics. It's true that there are ethical debates to be had around the diagnosis of single gene disorders where you can really trace what's going on in an individual. But what about a late onset disease with no cure? Would you want to know you were going to get a disease when you're a bit older? If you do decide to know, what about your family? Doesn't your diagnosis affect them? Supposing they don't want to know, what are the psychological impacts of this knowledge? Could there be genetic discrimination? But it's simply not going to be possible to devise simple genetic tests for intelligence or creativity. Thank goodness. It's just too complicated. I'm sure there'll be an explosion in internet companies offering such tests in the future. They should probably be banned, not because they might give you devastating information, but because they'll almost certainly give you misleading information. A probabilistic view, on the other hand, might tell you how best to avoid your particular personal risks. Part of me thinks, however, that the advice will boil down to needing to eat less fat, drink less alcohol, and take more exercise. Now, where have we all heard that before? I don't want to give you the impression, though, that genomics poses no social or ethical issues. There are questions of ownership and privacy and access, and certainly the need for debate. The debate needs to be had between well-informed stakeholders who understand the science, not lobby groups with a particular agenda. This is John Sulston, who masterminded the Human Genome Project. He unfortunately died rather unexpectedly earlier this year. And he worked tirelessly to make sure that genetic information was kept in the public domain, that genomes weren't patented and used by private companies for profit motives. And when the human genome sequence was first published, he said, we've now got to the point in human history where, for the first time, we're going to hold in our hands the set of instructions to make a human being. But I think that even more extraordinary than that is what has happened since. The technology for genome sequencing has developed so fast that it's now possible to sequence your genome for a few £100 in a couple of hours. But even more than that, than generating the information that's in our genomes, we can now very precisely edit our DNA using a technique called CRISPR-Cas9. So this technique, it makes use of the natural immune defenses of bacteria, which have come up with these molecular scissors. They're not really scissors, they're enzymes, but they remove unwanted DNA, and they evolved to remove unwanted DNA from bacteria. But we can use those to very precisely cut out sections of our own DNA with extreme precision. So you can guide these enzymes to cut the precise bit of DNA that you want to modify using a synthetic guide RNA molecule. We don't need to worry about the details of that. But when the cell repairs a break, errors can occur to generate a knockout of that gene. We can just remove that gene, or we can introduce additional modifications and edit that gene to change it slightly. So the technology, therefore, has the potential to rewrite the genetic code. What do we think about that? Genetic conditions could therefore be treated by modifying the DNA of affected cells, like rubbing out so many typos. If this base caused a disease like cystic fibrosis, well, we can rub it out. We can change that back to normal. So there's a possibility of radical new therapies for a range of diseases. Although these single gene disorders I've said are rare, if you add them up, there's actually affect an awful lot of people. So collectively, they're not rare. Genetic Alliance UK, for example, an organisation that represents patients with these Mendelian single gene disorders, estimates that 5.5% of the UK population suffer from one of these 6,000 easy to follow diseases, unlike the complex diseases that aren't easy to follow at all. So 5.5% of the population by the age of 25 will have one of these diseases. That's 3.5 million people. So the impact of genome editing in these patients and their families could be enormous. Human embryos could also be modified such that the disease-causing mutation is overridden, not just in that individual, but in their children and their children's children, and so on. This would be done by modifying embryos so early that any change would be incorporated into the germline, as we've seen, as the DNA replicates, it copies these mutations down the line. Is this a good plan? Could there be unforeseen consequences? Research institutions and funders and scientists have therefore called recently for renewed debate on the ethics of modifying human embryos before the science gets ahead of public opinion. The overwhelming view of scientists, I think, at present, is that the procedure is too new to know how safe it is. One big concern is that a public backlash could impact on the race to develop new and safe therapies. Is this playing God? Is editing the human germline a line that should never be crossed? Is it the first step on the road to these designer babies? Whatever we may think about the answer to this question, one thing is resoundingly clear. Talking about it is essential, like for genomic-based diagnosis. Public debate must happen, with all parties understanding the science, such that rhetoric doesn't get to replace fact. Public trust in science depends on openness, transparency, and effective regulation. And this is the approach that must be taken. There's no scientific answer to what is the right response to the potential use of a technology. That answer has to be provided by the whole of society. Many people wonder what makes humans so special. What are the special genes that we have that other organisms don't have? What sets us apart from other creatures? The truth is that there probably are very few genes that are distinctively human, unique to humans. No more than there are any particular genes that are unique to any particular organism. We might have a few more of one type of gene, a few less of another, a little tweak in the base composition of one gene here and there. But there's nothing really profound that sets us apart. And I kind of like that. Darwin said there was grandeur in his view of life, in evolution by natural selection. And I couldn't agree more. In fact, now we know so much more about the molecular biology and the genetics of all of this, that the view seems to get grander and grander. So we began with diversity in the natural world around us, and we're ending with diversity that you can see here. And that's what it's all about. Natural diversity being acted on by natural selection. But now we can create new diversity, and we need to think about what we think about that. But whatever we think about the future, one thing is definitely true. What a privilege we humans have in having evolved a brain advanced enough to both appreciate and start to understand this fantastic life all around us. Thank you for listening, fellow mutants.